So I just wanted to take a second to actually introduce your project for this unit. So you are starting this project quite early. You're starting the project before the Christmas break, and it won't be due to after the Christmas break, just because it requires so much setup. So what you're going to be doing in this project is you're going to be looking at the effects of either a biotic or an abiotic factor on growth. So for your lab itself, I actually gave you a lab organizer here. Please make sure that you use it because it's got all the sections of the lab that you need. So if you don't use the organizer, if you try to do your own lab report, there's a good chance you're going to miss one of the sections. So when you're ready to actually submit your final report, make sure you're using the organizer here that I provided for you. Make a copy. You can rename it whatever you want. And when you're ready, you're going to submit it to the Dropbox here. So in this case, we are going to be looking at grass. The reason that we're looking at growing grass is because grass actually grows within 3 to 4 days of first planting the grass seed. So you're going to plant your grass seed in class. Unless you're an online learner, then you come and you pick up your materials for your lab, and you're going to be actually growing your grass at home. So this is going to be a project that you're going to be doing at home over a period of about 10 days. So we're going to be looking at either an abiotic or a biotic factor that's going to impact grass growth. So just a reminder that an abiotic factor is a non-living factor. A biotic factor is a living factor. So you're going to pick one of these things. You're going to pick nutrients available, the amount of light available to the grass, the amount of water, the acidity of the soil, competition, or pests. Once you've picked one of those factors, you're going to actually design your own experiment. And during that experiment, you're going to tell me how this factor is impacting your grass growth. So you're picking just one of those factors, and you're telling me whether it's biotic or whether it's abiotic. Let's talk a little bit about how you're going to plan your experiment. So once you plan your experiment, you want to think about the one variable that you're going to change. So the variable you're going to change is going to be one of these. You're going to be changing nutrients. You're going to be changing the amount of light. You're going to be changing the amount of water. So you want to make sure you only change one variable. You want to tell me, is that variable biotic? Is it living? Or is it abiotic? Then you want to think about how you're going to observe grass growth. So I would recommend observing grass growth. You want to use any sort of quantitative measurement that you have. So I would suggest measuring the height of the grass growth. You have to decide how you're going to do that. Are you going to measure just one blade of grass? Are you going to measure multiple blades of grass and grab an average? Are you going to always measure the tallest? Are you going to always measure the shortest? How are you going to measure it? What tool are you going to use? So I'm going to leave that up to you. So you're going to pick your independent variable. It's going to be what you're going to change. You tell me if it's biotic or abiotic, and then you're going to go ahead and you're going to measure your dependent variable. You're going to be measuring your grass height. You want to make sure in your final lab report you include a purpose. So make sure that your purpose is what you're trying to study. So you want to tell me how you think your independent variable is going to impact your dependent variable. So how do you think changing the amount of water is going to change the amount of grass growth? By adding more water, is your grass going to grow higher? By adding more water, is your grass going to grow less? So you want to make sure you pick a purpose before you actually do the experiment. Once you pick a purpose, you want to write for me a hypothesis. Remember, you always write your hypothesis in an if-then statement. So if I change the independent variable, then the dependent variable will, and then make sure that you include a little bit about why you think this is going to happen. Why might water add to growth, or why might nutrients take away from growth? So you want to tell me why you think that is going to happen, why you've made that prediction. Once you've done that, now you're ready to actually go ahead and design your lab. So remember, you're going to be writing for me a procedure. Procedure is always a numbered list. You avoid first-person language, and in your procedure, you have to include both how you plan on planting your grass and also how you plan on actually making your measurements over your 10 days. You actually have to plant 2 cups of grass. So make sure you listen to this next part carefully. I want you to plant 1 cup. That's called a control. So what you're going to do is you're going to not do anything to that cup. You're going to add dirt. You're going to add grass. You're going to add a little bit of water. You're going to put it in that little Ziploc bag so the amount of water stays the same, and you're not going to touch it. This is how grass would grow under normal conditions. You call that the control. Then you're going to plant a second cup, and to your second cup, you're going to make your change. So let's say that you want to increase the amount of water. You would double the amount of water that you put into your second cup than you did into your first cup, and you do everything else the same. Same amount of grass seeds, same amount of dirt, and you put it in that plastic bag. If you want to limit the amount of sunlight, maybe in your second cup, you put a paper bag around it so it's not getting as much sun, or maybe you put it in a less sunny spot of your house. So you're going to have your control cup, cup number 1, and then you're going to have your actual changed cup, cup number 2, and that's going to allow you to compare how normal grass would grow and how the change that you made is impacting grass growth. Once you've designed your experiment, once you've written your procedure, you're ready to start collecting results. So you want to have an actual observation table. You want to measure your grass over a 10-day period. I actually put 12 here just in case you want to do 12, because oftentimes it takes 2 or 3 days to actually see grass growth. So you don't want these first couple days to be boring. So if you do have really no growth the first couple days, please try to get 10 days of actual measurements of actual seeing that grass grow. So you're going to have your 2 columns. You're going to have your control column, and you're going to have your change column. I want you to take whatever measurements you want. So again, if you're using a ruler, you're going to figure out in millimeters or centimeters how tall your grass is growing. You also, in your observation table somewhere, have to include pictures. This is so I know you actually grew your grass and you just didn't make up numbers. So you are getting marks for pictures. If you do not have pictures in your observation table, you will not get full marks. You have to make sure you include the pictures to show that you actually change the grass growth, or that you actually measure the grass growth. On top of that, you also have to include a graph. So I leave the graph up to you. You can use whatever graphing software you want. You can do a bar graph, a line graph, a pie chart, a scatter plot chart. There are a couple choices here that are better than others. So think about what you're trying to show in your graph. You're trying to show how the grass actually changed over the time period and which cup ended up with more grass growth. So you want to make sure that on your graph, you actually have both sets of cups. You have both the control and the modified cup, and pick which one you think is going to show that grass growth better. Make sure that you have a title, make sure that it's really clearly labeled so I know what I'm looking at. Finally, you want to write for me a conclusion. So in your conclusion, you're going to address a couple different things. You're going to tell me if your hypothesis was correct or incorrect, and you're also going to tell me why. So make sure that you include a couple data points about why it was correct or incorrect. Review what the independent and the dependent variable was. And then you also want to include any errors that you might have seen. Maybe you added so much water that your grass actually died, right? I want you to include any errors. If you didn't make any errors or your lab worked really well, think about what you were limited by, right? Is there anything that you were limited by, or is there anything that you could have done better? You're then going to go ahead and tell me how you could improve the experiment. So how would you fix those sources of error? Just a reminder that it does have to be in a report. You have to have all of these things in your report. So if you're following the organizer, then everything is there for you. And then to see how to get perfect, because that's what you want on this assignment, make sure you click on the rubric here, and it tells you exactly how the marks are allocated. If you have any questions, let me know. You do have to pretty much start this lab right away because it takes 12 days and it's due in a couple of weeks. So you can't leave this lab to the last minute. Otherwise, you're not going to have enough time to finish. So make sure that you sit down today or tomorrow, design your experiment, figure out what you're going to do, write your procedure, and get your grass planted. Good luck.